Hello everyone. My name is Wardhan Kulkarni and I am a first year MBBS student at Srimati Kashibai Navle Medical College, Pune. My topic for today is the urea cycle. I would like to break it down in short and simple 6 segments to make it easier. Starting with introduction. Urea cycle is also known as ornithine cycle or Krebs-Henselt cycle because it was the first cycle elucidated by Hans Krebs and Kurt Henselt. Urea cycle occurs in liver as there is one specific enzyme required in this cycle, which is arginase, present exclusively in the liver. There are total 5 reactions taking place in this cycle, which are catalyzed by 5 important enzymes, and there are 5 disorders related to the deficiencies of these 5 enzymes. There is also one syndrome related to this cycle, which is the HHH syndrome. Coming to the urea cycle. Urea cycle starts with the reaction of CO2 which comes from TCA cycle with ammonia. It gives carbamoyl phosphate under the presence of the enzyme CPS1, which is carbamoyl phosphate synthase 1. During this reaction, there is conversion of 2 molecules of ATP into 2 molecules of ADP. Carbamoyl phosphate then combines with ornithine, which comes from the other end of the cycle in the presence of the enzyme ornithine transcarbamylase and forms citrulline. Citrulline then combines with aspartate and forms arginosuccinate under the presence of arginosuccinate synthase. During this reaction, there is conversion of one molecule of ATP into AMP, so that means there is utilization of two high energy phosphate bonds. Arginosuccinate splits into fumarate and arginine under the presence of enzyme arginosuccinate lyase. Arginine then forms ornithine under the presence of the enzyme arginase. During this reaction, there is conversion of water molecule into urea, which is the product of the urea cycle. Ornithine then combines with carbamoyl phosphate under the presence of ornithine transcarbamylase to form citrulline, and the cycle goes on. So this is in short the cycle of the urea formation. Coming to the regulation of the urea cycle. Urea cycle is regulated by two means. First, the allosteric regulation, and second is compartmentalization. N-acetyl glutamate or NAG acts as an allosteric activator of the first enzyme of urea cycle, which is CPS1 or carbamoyl phosphate synthase 1. The first two reactions of the urea cycle take place in mitochondria and the next three reactions take place in cytosol. This is known as compartmentalization. So these two factors contribute to the regulation of the urea cycle. Coming to the energetics of the urea cycle, I'll divide it into two parts. First, ATP utilized. There are two reactions in which ATP is utilized in urea cycle. Reaction 1, that is conversion of CO2 and ammonia into CPS1. In this reaction, there are two molecules of ATP consumed. Third reaction, conversion of citrulline into arginosuccinate. In this reaction, there is utilization of two high energy phosphate bonds. So, in total, there are 4 ATP consumed. Coming to the ATP gain, in the conversion of arginosuccinate into arginine, there is also formation of fumarate, which undergoes TCA cycle to form NADH, and that NADH, when undergoes oxidative phosphorylation in the inner mitochondrial membrane, forms 2.5 ATP. So the ATP formed are 2.5. So the net ATP consumed would be 4 ATP which are utilized minus 2.5 ATPs which are formed, so 1.5 ATP. So as we have discussed earlier, there are 5 enzymes. So there are 5 disorders related to the deficiencies of the 5 enzymes. Coming to the first enzyme, which is carbamoyl phosphate synthase or CPS1. CPS1 catalyzes the conversion of CO2 and ammonia into carbamoyl phosphate. So the deficiency of CPS1 would lead to increased levels of ammonia, which is known as hyperammonemia type 1. Same goes with ornithine transcarbamylase. Ornithine transcarbamylase deficiency leads to hyperammonemia type 2. Arginosuccinate synthase catalyzes the conversion of citrulline into arginosuccinate. So deficiency of this enzyme leads to increased levels of citrulline, which is known as citrullinemia. Arginosuccinate lyase catalyzes the conversion of arginosuccinate into arginine. So deficiency of arginosuccinate lyase leads to arginosuccinate aciduria. Coming to the last enzyme, which is arginase, which is exclusively present in liver. Arginase catalyzes the conversion of arginine into ornithine. Deficiency of arginase leads to increased levels of arginase, hence causing hyperarginemia. Coming to the last part of this presentation, that is one single syndrome associated with this urea cycle, which is known as the HHH syndrome. HHH syndrome includes the following three disorders: hyperammonemia, hyperornithinemia, and homocitrullinuria. The major cause of this syndrome, HHH syndrome, is due to the defect in ornithine transporter 1G. With this, I would like to conclude my presentation and thank each and every one of you who have watched this video till the end. Thank you.